Skeletal muscle consists of bundles of tiny myofibrils that run the length of the muscle. Myofibrils consist of even finer fibers called myofilaments. They are arranged in a type of latticework to form units called sarcomeres. There are two types of myofilaments, thin and thick. Thick myofilaments are made of a protein called myosin, whereas thin myofilaments consist of a protein called actin. Thin myofilaments are attached to Z-discs. When a muscle begins to contract, the myosin heads latch onto the actin myofilaments, forming what is known as a cross bridge. The myosin heads propel the actin myofilaments forward toward the center of the sarcomere. Because the actin myofilaments are attached to the Z-discs, they pull the Z-discs closer together, shortening the sarcomere. As the sarcomere shortens, so does the myofibril and the entire muscle. Keep in mind that the myofilaments do not shorten. They stay the same length. The sarcomere shortens because the filaments slide over the top of one another. A skeletal muscle must be stimulated by a nerve, specifically a motor neuron, to contract. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction. Between the end of the motor nerve and the muscle fiber is a narrow space called the synaptic cleft. When an impulse reaches the end of a motor neuron, it causes small vesicles to release a chemical messenger or neurotransmitter called acetylcholine into the synaptic cleft. The acetylcholine quickly diffuses across the synaptic cleft, where it stimulates receptors in the sarcolemma, the membrane surrounding the muscle fiber. In turn, this sends an electrical impulse over the sarcolemma and inward along the T-tubules. The impulse in the T-tubules causes the sacs in the sarcoplasmic reticulum to release calcium. The calcium binds with the troponin on the actin filament to expose attachment points. In response, the myosin heads of the thick filaments grab onto the thin filaments and muscle contraction occurs. When nerve impulses stop arriving at the neuromuscular junction, acetylcholine is no longer released. Calcium ions are pumped back into the sarcoplasmic reticulum. With the calcium removed, troponin and tropomyosin again prevent the myosin heads from grasping the thin filament, and the muscle fiber relaxes.